Hi everyone, I'm Zi Xuan, a PhD student from Aerobic lab. The literature I want to share with you was published in Nature Neuroscience. Aerobic glycolysis is the predominant means of glucose metabolism in neuronal somata, which protects against oxidative damage. Firstly, let's have a look at the overview of the research. Here is a neuron schematic figure. The neuron is a highly polarized cell with a central soma that contains the nucleus, more distal processes, dendrites, and axons that generally receive and transmit synaptic signals. While there are differences in levels of aerobic glycolysis and mitochondrial oxidative phosphorylations between the somata and the terminals remains unknown. Thus, the researchers performed metabolic flux experiments as a starting point for exploring whether glucose metabolism is mediated via a distinct mechanism between the somata and nerve terminals. They observed that neuronal somata exhibit increased aerobic glycolysis and reduced OXPHOS level relative to terminals in both basal and activative states. This discrepancy is associated with the predominant localization of the glycolytic enzyme pyruvate kinase 2. Deletion of Pkm2 in mice induces a shift from aerobic glycolysis to OXPHOS in neuronal somata, resulting in oxidative damage and the gradual loss of dopaminergic neurons. Their findings update the conventional view that neurons uniformly use OXPHOS under basal conditions and highlight the important role of somatic aerobic glycolysis in maintaining antioxidant capacity. Now let's go deeper to the story. I'd like to start with the introduce about some background. OXPHOS, mitochondrial oxidative phosphorylation, is a metabolic pathway in which cells use enzymes to oxidize nutrients, thereby releasing chemical energy in order to produce ATP. Almost all aerobic organisms carry out oxidative phosphorylation. This pathway is so pervasive because it releases more energy than alternative fermentation processes. Although oxidative phosphorylation is a vital part of metabolism, it produces reactive oxygen species, such as superoxide, oxide, and hydrogen peroxide, which lead to propagation of free radicals, damaging cells, and contributing to disease and possibly aging and senescence. Aerobic glycolysis is a metabolic process by which cells metabolize sugars via fermentation in the presence of oxygen and occurs through the repression of normal respiratory metabolism. It allows proliferating cells to convert nutrients such as glucose and glutamine more efficiently into biomass by avoiding unnecessary catabolic oxidation of such nutrients into carbon dioxide, preserving carbon and carbon bonds, and promoting anabolism. Aerobic glycolysis redirects pyruvate generated by glycolysis away from OXPHOS to generate lactate. Is another approach to reduce excessive RS produced by OXPHOS. The disadvantage of aerobic glycolysis is its inefficiency in ATP generation. Aerobic glycolysis produces only 2 ATP molecules per glucose molecule, whereas OXPHOS can produce up to 36 ATP molecules after complete oxidation of 1 glucose molecule. And here is the Pkm2. It can promote the reduction of pyruvate to lactate. Speaking of neuron, it has extraordinarily high energy demands compared to other cell types due to their specialized functions, such as the maintenance of membrane potentials, neurotransmitter release, and recycling and exoplasmic transport. The conventional view holds that under basal conditions, neurons generate ATP predominantly through OXPHOS. And here is a previous work has demonstrated that neurons have a lower glycolytic rate than astrocytes and sustain antioxidant protection via the use of glucose through the pentose phosphate pathway. So, considering that glycolysis is another approach to reduce excessive loss produced by OXPHOS, and the neuron is a highly polarized cell, they question that whether there are differences in levels of aerobic glycolysis and OXPHOS between the somata and nerve terminals. They isolated neuronal somata and synapsomes from the whole brain to investigate differences in glucose metabolism using metabolic flux and cellular respiration analysis. Subsequently, they incubated isolated somata and synapsome using isotope-labeled glucose. Besides, they cultured isolated somata and synapsomes with 2-deoxy-D-glucose. These results demonstrate that glucose uptake and glycolytic flux are higher in the somata than in nerve terminals, and the reduction of pyruvate to lactate is greater in the somata than in nerve terminals. Because the isolation procedures may impair the metabolic activity of subcellular preparations, and the minor contamination of glial cells is inevitable, they further explored the differences in glucose metabolisms between somata and nerve terminals in vivo. Using genetically encoded sensors combined with miniature 2-photons microscopy, they compared OXPHOS and glycolytic contributions to ATP levels in awake mice. As the results shown in this figure, the cortical neurons perform higher level of aerobic glycolysis in somata than in nerve terminals. After a clean way to prove the differences in levels of aerobic glycolysis and OXPHOS between somata and terminals, and then they next to study further, for example, like the protomic differences. They analyzed the protons of neuronal somata and synapsomes using tandem mass tag-based quantitative proteomics to investigate the molecular basis for the metabolic differences between somata and terminals. The results of KG gene enrichment analysis also demonstrated significant differences in several synapses-related pathways between somata and the terminals. In the background, I mentioned that aerobic glycolysis redirects pyruvate to generate lactate. It is an approach to reduce excessive loss produced by OXPHOS. In the pyruvate is produced from PP under the catalytic action of pyruvate kinase. There are two main isoforms of the Pkm enzyme exist in the brain, Pkm1 and Pkm2. Previous studies have shown that Pkm1 promotes the entry of pyruvate into the PCA cycle, and Pkm2 promotes the reduction of pyruvate to lactate. Further, they use the TSA multiple staining to detect the distribution of Pkm1 and Pkm2 in the somata and nerve terminals in various brain regions of mice to explore the differential distribution of Pkm1 and Pkm2. They found that levels of Pkm2 are higher in the somata than in terminals, and Pkm2 is prevalent throughout the whole brain. in their supplementary results. After getting these results, they questioned that whether high expression of somatic Pkm2 determines the high degree of aerobic glycolysis in somata. They conditionally knock out Pkm2 in SSP area to explore this question. By two-photon microscopy, they found that following Pkm2 deletion, inhibition of mitochondrial complex 4 by oligomycin A caused a more notable decline in ATP levels in somata, suggesting a shift in somatic glucose metabolism from aerobic glycolysis to OXPHOS. They further investigate why the Pkm2 drive high aerobic glycolysis in the somata in other brain regions. Then they conditionally knock out Pkm2 in SNC and detected changes in glucose metabolisms in the SNC region. As shown in the figure, Pkm2 deletion led to a decline in levels of G6P and lactate, and an elevation in levels of TCA cycle intermediates in the SNC region. Similarly, KEGG metabolite enrichment analysis showed a significant change in the TCA cycle. These results suggesting that Pkm2 deletion in dopaminergic neurons switched the somatic glucose metabolism from aerobic glycolysis to OXPHOS. After getting these results, they want to understand the reason underlying high aerobic glycolysis in somata. The special metabolomids demonstrated the results that they found that a remarkable reduction of antioxidants reduced aerobic acid after Pkm2 deletion, indicating that the alteration in somatic glucose metabolism impaired the redox buffering capacity of neurons. Hence, they speculate that somata perform high aerobic glycolysis to prevent oxidative damage. To test their hypothesis, they detected the expression of 4-HNE produced by lipid pre-oxidation and GAM-H2AX, a biomarker of DNA double-strand breaks in cortical and dopaminergic neurons after Pkm2 deletions. In dopaminergic neurons, both lipid and nucleic acid oxidative damage increased after the somatic switch in glucose metabolisms. These results indicate that somatic glucose metabolism alterations in different brain regions can lead to oxidative damage in neurons, suggesting that somata use high aerobic glycolysis to prevent oxidative damage. Speaking of oxidative damage, it is one of the major factors inducing dopaminergic neuron loss in Parkinson's disease. Hence, they aim to detect the loss of dopaminergic neurons after Pkm2 deletion using the immunohistochemistry method. As shown in this figure, there was a significant loss of dopaminergic neurons compared to the when Pkm2 is deleted for 3 months. These results indicated that the alterations in glucose metabolism in the somata cause progressive loss of dopaminergic neurons. So finally, let's summarize this story. In this study, they reported a remarkable metabolic divergence between somata and terminals, with somata displaying heightened levels of aerobic glycolysis and reduced OXPHOS activity. This metabolic shift, as they demonstrated, is mediated by the Pkm2 within somatic regions. Furthermore, their experiments involving Pkm2 deletion in mice unveiled a consequential switch from aerobic glycolysis to OXPHOS in neuronal somata, leading to oxidative damage and the progressive loss of dopaminergic neurons. Their research updates the conventional concept that OXPHOS is used for metabolism throughout all neuronal subcompartments and basal conditions. In their original literature, the more data in the supplementary results. If you are interested in this paper, you can find it and read it thoroughly. If you have more questions, feel free to ask me. That's all. Thank you.